Yeah, we'll start the class. Good evening. Good evening, everyone. Today I'm going to discuss about the extension part of the CRISPR-Cas9. Please watch my video. Like, if you not watch, sorry, not my video. Like, what the link I have shared in the group. Please watch the two videos. Like, if you not understand today's class, so I will be giving glimpse, and you just watch the video. You will get an idea like how these things can work. Okay, we talked about what is CRISPR-Cas9, what is genome editing, types of uh, what is genome editing, and what are the different genome editing technologies are available, and which one is famous today, and how we can introduce this construct into a plant, and how we can do genome editing, and how we can isolate the plant without any transgenic, but only with the desirable mutations. We have discussed in detail about all these things, and we will take some questions first, then we will move towards it. Okay, which one of the following is a template guided repair target DNA break using a sequence template containing an entire gene or even a longer genetic elements? Which one will be the answer? Can anyone guess?
SDN3, sir.
And three. Why?
In that only we will introduce a whole gene or even a long genetic element.
Exactly. Okay, but rest other just either we allow for a mutation by itself, like repair by itself, either insertion and deletions, or we can give a small template having 2 to 3 nucleotides in SDN2. But in SDN3, we are introducing entirely a new gene into the genetic background of the plant. That's why SDN3 is your template guided repair, where entire gene or even longer genetic elements can be introduced. Okay, SDN3. Then here same question repeated like it is in 2023-22. SDN2 involves template guided repair of a target DNA break using an externally supplied template sequence, SDN2. Then SDN3 involves template guided repair of a target DNA break using sequence template, typically double-stranded DNA containing an entire gene or an even longer genetic elements.
Both one
Both
and
are
two are
true.
true, sir.
Both are true. Okay. Then we discussed. We have discussed, which is not a generating self-discuss. See, this again, two questions came last year. Okay. It's very, very important. This one and this one. Respect. Very important. 10 marks. Then this year they were asked about base editing. With a simple definition only they were asked. You know that what is the definition of the types of with different mechanisms, and we will wind up. Okay. Discussed about what is CRISPR-Cas9 based editing. Scientists thought that here, while looking at the Cas9 structure, it's a protein, so that we can modify this protein can be used for multiple purpose. Either you can use it for uh, either you can use it, you can modify this particular protein. Either it can be used for either base editing, prime editing, or you can use it for epigenome editing or genomic suppression, whatever it may be. There are a number of uses. Like I will share on PEP. Like we watched the video CRISPR and Beyond. Like in that video, it's detailedly explained what are the additional technologies developed based on this CRISPR-Cas9. And today we are going to discuss only important technologies called as base editing and prime editing. And there are several RNA editing also there. Please read the paper. I will share that. Okay. Today I'm going to cover only base editing, prime editing, and we'll jump into the RNA silencing. Then, as I earlier discussed, the Cas9 structure having two domains, RUVC domain and HNS domain. What it will be do like HNS domain is in target and RUVC in non-target. You can see where your single guide RNA come and bind to the strand, it is called as target strand. Where other strand which is complementary to this strand, where your RNA will not go and bind, that is called as non-target strand. Here you can see that RUVC domain cuts a non-target strand and HNS domain cut target strand. Please remember, it asked in so many exams like CSIR gate. Okay, RUVC domain of the Cas9 protein will cut non-target strand, but HNS domain will cut target strand. You can if you mutate any of this sequence, RUBC or HNH, your double stranded break will not happen. For example, sorry, uh, break will not, double stranded break will not happen. But if you are mutating only RUBC, you are allowing HNH, means only target strand in the break will occurs in target strand. If you are mutating HNH, but your remain like your RUBC is inactive, it will cut only non-target strand. Either you can create mutation in target only by mutating HNS domain, sorry, non-target only by mutating HNS, or we can allow mutation in HNS domain by sorry, target like a target strand by mutating HNS. Either you can do. But idea is instead of double stranded break, you can introduce only single stranded break either of the any strand by mutating any of these nucleus domains of Cas9. Understood what is this? RUBC domain, HNN domain, HNS domain, and target and non-target strand.
Yes, sir.
Yeah. Then this is called as Ncas, like Nicase. For example, if you're mutating any of this domain, then it is called as Ncas9. Like it will become a Nicase. It will not become a double strand nucleus, but it will just nick the strand. Means it will create the nick in any one of the strand. That's why it will become Nicase. By using this one, there is a one strategy called as base editing. Means it's a base editing is it's one of the genome editing approach where it can convert one DNA base into uh, another DNA base without introducing a double-stranded break. We will see how it will work. Means what are the components of this base editing is it having critically catalytically impaired Cas9 protein. Means one of the domain is mutated. Both the domain is not active. Any one of the domain is mutated so that its catalytic activity is impaired. Then it having at Cas9 it is fused with an enzyme called as deaminase. What is the function of deaminase enzyme?
Remove the amino groups, I think.
Yeah. Then you can use uracil glycolase inhibitor. Means what is the main function of this one? It prevents the cellular repair mechanism from reversing the base conversion. What it happens? For example, you are changing one base to another base. Definitely, there will be a repair mechanisms in the host. What will they will do? They will come and if you creating any modification, they will come and they will again repair them. But you don't want that. That's why if you add uracil glycolase inhibitor, so that what are the modification you make, it remain intact. Means it suppresses the host repair mechanism so that what are the modification you done, it will be remain intact. That is the idea behind why we are using uracil glycolase inhibitor. Remember, there will be three components. One is your Cas9 is mutated, called as Cas9 nicase, or it can also called as dead Cas9, where Cas9 nicase only one of the domain is mutated. If dead Cas9 means both the domain, like RUBC and HNS domain is mutated. But if any one of the domain is mutated, it is called as nicase. Both the domain mutated is called as dead Cas9. Means the function is the nucleus function is completely uh, eliminated. Then the another part is deaminase enzyme, so that this enzyme will remove amine group from any of the bases which are you are targeting. Then it will, if you remove an amine group, there will be change in the structure. Then there will be change in the nucleotide. We will discuss what is the famous if you like uracil to cytosine, cytosine to uracil. Like that, there will be changes will happen by using this deaminase enzyme. To avoid to suppress host repair mechanism, we are using uracil glycolase inhibitor. Then, what type of base editing can we do? Either there are two types. Cytosine base editor, you can modify the cytosine or you can modify the adenine. Means here C can be converted, C to G pairing can be mutated to T to A pairing, or A to T pairing can be mutated to G to C pairing. How we can do CG to TA? by deaminating the cytosine to uracil, which can be recognized as a thymine during DNA replication repair. Okay, if you deaminate the cytosine to uracil, if you deaminate the cytosine, then it will become uracil. Then definitely uracil is paired to thymine. But cytosine normally should pair to what? Cytosine should be paired to
Guanine.
guanine. But if you're removing amine group from the cytosine, it will become uracil. You know what is it? It will become uracil. But uracil counterpart is uracil paired to thymine. Instead of CG, you are making it to thymine, you are by converting into uracil, then thymine, then mutation will occur thymine, then in next replication, then definitely your T will pairs to A. Instead of CG, you are bringing TA. This is the by that this mutation can be done by using your base editing technology. Then and one thing, we are not making any double stranded break. Either we nick by using nickase, either you can use decase without any double stranded break, we can modify. Because we are adding deaminase, only we need guide RNA. For example, you want to targeting guide RNA here, use it for targeting. For example, you want to make mutation here, but how to reach here? Only by your guide RNA and this Cas9, your whole CRISPR complex will come and will bind here. Okay. After binding, you just want to modify the base. That's why we are mutating both the or one of the domain or both the domain of Cas9, so that we're not introducing any double stranded break, but we'll modify the particular. For example, if C is there, you'll deamine it will be read as a uracil, then thymine will incorporate, then T2A mutation will occur. Like this, we are modifying the bases without having any double stranded break in the DNA. Okay, understood what is base editing? This only asked.
Yes.
If you are fusing deaminase enzyme with the Cas9, which is the technology, base editing, the primary, there are given several options, but you should opt this one only. Then you can also modify adenine by using adenine base editor. You can modify AT to GC. Means if you are deaminating adenine, it will become INSC, which actually interpreted as guanine. Then definitely next cycle guanine again cytosine. Means adenine should be paired with thymine, but you are modifying the adenine in such a way that it can be regarded as INSC. Then it can be regarded as guanine. Means guanine cytosine will be taken. Means AT to GC, you are making changes by using deaminase enzyme. Okay. Either you can use cytosine base editor or adenine base editor. We are introducing CG to TA mutation or AT to GC mutation by using these aminase enzyme along with your Cas9 nickase or decase9 along with your guide RNA. This is what the base editing looks like. Okay, without introducing double stranded break. Understood what is base editing? You will see
Yes,
in
sir.
figure. It will be very clear. See, instead of your Cas9, you are having N-Cas9 and it is fused with cytidine deaminase. Where you want to change this one, it will like along with uracil guanidine. What is uracil glycolase inhibitor and your cytidine deaminase along with N-Cas9. You are targeting DNA and you are modifying the base. See C to A. Like this, we are introducing or modification by using base editing. Okay, without introducing any double stranded break. See here, no double stranded break will happen. Then another technology we will discuss, that is called as prime editing. See here only we can have transition, like either you can have only two possibilities are there. But if you want to introduce a series of nucleotide, for example, AAT, you want to introduce 3 nucleotide at a time into genome, how we can introduce? With normal Cas9, we can't do this. With base editing, we can't do this. That's why scientists designed another technology called as prime editing. You understood why? You understood this point? If you
Sir,
want to
with S,
introduce what? Yeah.
sir,
If you want to
with
introduce a
SDN2
set
we
of
can
nucleotides, what?
with SDN2 we can't introduce this This short sequence AATT.
No, without providing any template. Like without inserting this particular this one into the see in template within three only we can't do. Like we can do, but without providing external template, we can do this one.
Okay, sir.
Then you watch that video, it will be very very clear. Okay, like here may you not get. I think while I am reading at first time, like in my classes I not understood. After watching animated video only, you'll get. Like prime editing is difficult to understand actually when compared to base editing. Base editing is easy, just adding BM minus and it will modify the base. Prime editing, you just watch the video, like how pegRNA will come, how it will bind to the RNA of one strand, then again how transcriptase we'll discuss. Then actually it is more versatile genome editing method. Means either you can allow precise insertion or deletion, all possible base substitution without requiring donor template. Okay, here this point is very very important. Even though don't have any template, you can introduce precise insertion and deletion to the genome. How we can do, we will discuss. Okay. Means there are two major components. Prime editor protein, like it's composed of Cas9 nickase fused with reverse transcriptase. Please remember in prime editing you are using reverse transcriptase, Ncas9 along with pegRNA. Prime editing guide RNA. Three, remember, either mutated Cas9, reverse transcriptase, pegRNA. Then modified guide RNA consists of having targeted sequence, then template for desirable edit, and primer binding site for reverse transcriptase. Okay, means you are having a reverse transcriptase means you should have some primer, no? Because it's a it is bind to your DNA, means where the primer is there, then only it will bind. Then you should have this RNA provides the template to bind the transcriptase enzyme, and then it will also bind the target strand. Then your process will occur. We will discuss how exactly. First, Cas9 will come. Cas9 will create single stranded DNA nick at target site. Then your pegRNA will direct the reverse transcriptase to the nicked strand. Then reverse transcriptase synthesizes the new DNA sequence based on the template pegRNA, and cellular repair mechanism incorporate the edited DNA strand into the genome. We will see the video so that we will get in detail. Wait, I will share the screen. Okay, my screen is visible now.
Yes, sir.
Hello. Just watch this so that we'll pause and we'll explain. You have a target here, and definitely your guide RNA will bind only when adjacent to your PAM site. As I told, like we are using Ncas9 so that introduce instead of double stranded break. Here we are creating the nick, and see here we are creating the nick in which strand, target strand or non-target strand.
Target strands.
Wait, okay. See, I told which is target and which is not target. We will discuss. Right. See, then actually your mRNA will bind here. The target of mRNA is this one. This one will be the target. And you are introducing the nick in non-target strand where if we are which of the domain of Cas9 is introducing introducing nicks in the this break in the DNA is which NH
RUBC,
are
non-target strand.
yeah, okay, this nick is caused by RUBC. Then where nick is there, left part of this one, my arrow mark is visible.
Yes, sir. Yes, sir. Visible.
Yeah, your left part will provide, it is for your left part. We are designing such a one part is called as, we are designing peg RNA in such a way that left to the nick, it should provide you should it will bind to the template where it having sites for your transcriptase. You will watch this. Understood? Any doubts? Understood or not? This is very
Sir,
simple. You watch it again so that you will get it. Okay?
sir, how that the original strand got removed, sir?
Which original strand?
Mm. We created one reverse transcribed DNA, sir,
Yeah,
and in
actually
that
it's a
place,
flap. Flap. Actually, that is whenever when it is reaches na, like you know that in eukaryotic DNA replication, after there will be flap removal. When it is, you will watch clearly. See, what happens? Like it will create the flap na. Then based on that one, after when it is reaches and flap will arise, then definitely then your because of that flap will, whenever the flap will arise, one particular mechanism it will remove that flap so that your original DNA strand will be removed. See, equilibrium between the two flaps. When actually it will arise above, then it will cut. The flap will be cutted. Like flap endonuclease enzyme you heard in eukaryotic DNA replicase. In the same way, it
yes,
is
sir. Mm.
functions. Okay. Okay. This is what this is very simple, but you just remember what are the components at least we are using. We are using PEG RNA and Cas9 and reverse transcriptase. And we are introducing several repeats like insertion, deletion that cannot be possible in the where normal Cas9, and without template we are introducing this. If you want to introduce a particular sequence or a gene, we need a template. But sometimes we don't have template means, or because you know that if template guided repair should happen, which stage it is frequency is very less. It should have happens only in a space of the life cell cycle. But with the prime editing, we introduce some set of nucleotides like insertion, deletion, or a series of nucleotides like AATT or TTA. Any stage of the cell cycle. It is independent on like it's not to be like it's only on a space. It is not like that. At any stage of the cell cycle, you can introduce this one, your desired strand. That's why it is more useful. Okay. Then it contains reverse transcriptase, nickase, and peg-RNA. Like in video, how first Cas9 will create single-stranded nick, then your peg-RNA will direct the reverse transcriptase to nick the strand. Uh, then reverse transcriptase synthesis a new DNA strand based on that, like based on the template that you are providing by peg-RNA. Then again cellular repair mechanism like flap endonuclease. This will cut the this one strand and it will join and it will ligate. So that you are introducing your desired mutation in uh, in a genome. Understood? Please watch it again because I know it is takes time to understand. Just watch and like there are better videos than these videos, but it's a very short and neat video. That's why I showed that video. Either Nature's one video is there, I shared. You watch that one, it is completely animated video that you can watch. You will get an idea. But you should remember components at least. Should I reverse transcriptase enzyme, peg-RNA, nickase, Cas9 nickase. Okay.
Okay.
Then like having N-Cas9, peg-RNA, and reverse transcriptase. Wherever you nick left strand side, it will provide. It will having a primer to. It will primer sequence to that where your reverse transcriptase will bind and it synthesize the template strand and it will introduce the nick. Then wherever the flap will come, then actually it is a. I'm showing that part, the mechanism like in the video. Then you are introducing your desired. Okay. Either you can deletion up to one base pair to more than four base pair you can do. Either insertion up to 80 base pair you can do with the prime editing, but you can't do in base editing and normal editing without providing any external template. That you should. Then what are the advantages? It's allow all possible base substitutions and enable small insertion and deletion like indels you can introduce, and this does not require any double standard DNA breaks. Means if you are introducing double stranded DNA break, sometimes what will happen? We are getting unwanted mutations while repair. That we can avoid. Then it is more flexible and more precise than the base editing. Okay, these are the advantages of prime editing. Then please, for our exam like SRF, they will ask just definitions and the components because it's more biotechnology based. For biotechnology people, they will ask in detail. In CSIR exam, they will ask in detail. But for SRF, for ASRBNet, you just remember definition and the component. It's more than enough. Okay. Then what is why we need genome editing? Like we can introduce precisely targeted gene mutations. Apart from this, they have several advantages. And please read papers regarding to the each heading. I'm just giving the heading here. Please read all the headings. Okay, there are several papers. Uh, you just read like what is de novo domestication. That it is very important actually. Then you can have a directed mutagenesis that we have seen where your conventional mutation is random in nature, is undirected. That is one of the advantage. And nowadays we are doing with a multiplex genome editing. Means, for example, at a single time we can edit more or the like more than one gene. We are talked about we are editing about one gene. All these base editing or prime editing or your CRISPR-Cas9 editing, it is editing only single gene. But at a time, you are doing all your you are creating a construct, you are introducing your regenerating plant only with one gene. Why you are doing? We can modify or you can edit multiple gene at a single time in a single construct instead of a single guide RNA. If you introduce multiple guide RNA in a single construct and you introduce it to plant and you can edit multiple genes at a time, that is called as multiplex genome editing. Okay. Like for example, like you can introduce modification of different genes by providing a different guide, single guide RNA strand in a single construct and we are editing all the genes. That is called as multiplex genome editing. There are like Cas9 based multiplex editing, Cas12 based multiplex editing. I'll share the paper. Please read that. It's very easy. Okay. Then you can able to edit quantitative trace loci. See, first we have to take the what are the number of alleles, and if you are which allele is having more effect, that after identifying that then you can identify the particular allele so that your expression of QTL will be increased. That you can do. Then de novo domestication. What is the de novo domestication? Normal domestication, it will take longer time. You want that trait. For example, you having taller plant height, like say wild diary, but you are making into short plant stage. But it will take normal domestication very long years. It take hundreds of years. But we can accelerate domestication by mutating or by editing some domestication genes. That's why, like, it will take around 2 to 4 years. We can domesticate any particular trait. This is called as de novo domestication or accelerated domestication. Please read this paper. Like, you just type de novo domestication or accelerated domestication. Several papers will come. You read any of the paper. Then we can induce the like haploid induction, and also we can create artificial apomixis. Like nowadays, there is a the term called as synthetic apomixis. Means there is a one particular gene is called as two particular, either baby boom, BBM means baby boom, or MIME gene. Means editing this gene without a contribution of the male parent, we can stimulate the egg development, like fertilization. For fertilization, the pollen should be there and the male gamete should come. Then after fusing with the egg, your fertilization will start. Then scientists thought like, why the male gamete is needed? What's there in the male gamete that's stimulating the egg to fertilize and forming the embryo sac? What forming the zygote? What is the contribution of male? They identified that there is some transcription factor which actually responsible for stimulating fertilization in the egg. Actually, they introduce them, but that is there in the female itself, but it will cause some mutation. That mutation is created by gene editing, so that without contributing the male parent, we can stimulate the fertilization, so that we can develop a homotic seed. That can be also done. Either I told like SNH3 gene is very very important for means you no need to cross, no need to maintain, no need to isolate. You just in the host itself, you modify that particular gene. There will be definitely there will be segregation imbalance if you modify this CNH3 gene, like meiotic cover, there will be uh, meiotic, the segregation distortion will be there. Then you can induce the haploid by this. There are again several beneficial advantages like large scale screening of entire genome wide mutations. Then synthetic biology means you can introduce a new pathway into the plant by using your CRISPR-Cas9 technology. Nowadays, we can modify the plant microbiome surrounding the rhizosphere so that we can re-engineer that microbiome by gene editing technology so that we can improve the plant and microbial interaction. Anyways, you can it, like genome editing play several roles. Not only this, like de novo domestication or directed mutagenesis, increasing the expression of QTL and induction or synthetic apomixis or synthetic biology, plant microbiome engineering. There are a lot of things, sir. We can also suppress the epigenetics, like epigenetic editing by CRISPR also there, or CRISPR mediated gene silencing is also there. There are several other advantages also there. Please read it. It's very, very important for exam. Okay. Understood what is base editing, prime editing, what are the different advantages of CRISPR-Cas9? Any doubts regarding it? Hello.
Sir, all these are important for sir.
Yeah, sometimes they may ask. Okay. Because we can't guess what they will going to ask. But more into the biotechnology, don't go. But you just take, for example, this haploid induction and artificial apomixis through genome editing. It's very important for us because it's used in the plant breeding and editing the quantitative trait loci. It's important for the plant breeding. De novo domestication. It's important for plant breeding. In that way, you have to read. But plant microbiome engineering is not needed for us. Don't go in detail. And synthetic biology also not needed that much for us. You just leave it. But you should know what is synthetic biology. Like we are introducing a new path. For example, a classical example of synthetic biology is, can anyone tell? Golden rice. We are introducing the vitamin beta carotene synthesis pathway in the rice. It was earlier, it was not there. Okay, so that we are increasing vitamin A. That is a classical example of synthetic biology. Okay, that's why how we can introduce a different pathway, or you can modify the pathway in such that beneficial for us by genome editing, that we should remember. But at least read the articles regarding de novo domestication or haploid induction through genome editing. Okay, and multiplex editing is not needed. Just you can you can edit the different gene at a single time by using Cas12 or Cas9 modified Cas9. That you can do. But rest other thing is not that much important. But as a breeder point of view, how we can use the genome editing, we should use. And before ending this class, I want what prerequisite we need for editing to successful gene editing in case of applied plant breeding, like applied genetics, plant breeding. Main thing you need trait. Sorry, first you need variety. First you have to select variety. Which variety you are doing genome editing? Normally, which variety we choose for genome editing? I'm thinking like you are a breeder. Normally, which variety you want to create like mutation or editing? You want to do editing as a breeder? Don't think like as a basic biologist.
Best performing varieties.
Yeah, best performing variety or mega varieties. Like in Samba Masuri, it's a classical example. Samba Masuri or PPT 4004 something. Samba Masuri. It's a well-known variety. So a highly exported variety after Pusa Basmati. It's a like non-Basmati. It's high exported variety. Till now it's a farmer favorite variety. Then you have to choose varieties so that it is already there in the farmer's field in a larger scale. It should be a mega variety. Then you should do SWOT analysis of this variety. Strength, weakness, opportunities, and threats. What is the strength of this particular variety? You should remember. What is the strength of the Sama Masuri? We discussed.
The green type, it's green type, medium slender, high
Yeah,
yielding.
medium strength, stature, and high yielding. It's cultivated in a larger scale. Farmer's favorite. And farmer don't want to replace. This is the strength of this variety. What is the weakness of this variety? Means it is, even though it is having high yieldings, but it having susceptible to several biotic and abiotic stress, heat stress, drought stress, whatever it may be, phosphorus use efficiency. What is the opportunity we can able to increase the yield further, and we can overcome this all biotic and abiotic stresses? Then, and another, what is the threat? For example, if you are modifying something, you should not limit the other trait. For example, it's a medium stature and something you take, or culm strength, or you take, or grain characters. For example, you are modifying any grain size. Means ultimately you are compromising grain character. Means smaller grain, it is most preferred in South India. Smaller, shiny grain. But if you are increasing grain size, definitely it is not preferred by the farmers in South India. Then what? If you are editing smaller to larger, what will happen? Farmer preference will be going to change. That is a threat. You are editing something means you should take care of that. You are not uh, compromising any other trait which are preferred by the farmer. That threat will be there, that you should take care. First, you do SWOT analysis of the particular variety. Then you decide where is the opportunity to edit, and you should also take care of what threat you are going to face and how to overcome this. That first thing you should remember before editing. Then second thing, first one is selecting the variety. Then next thing what you can expect,
We need the sequence information.
before that, first you choose a variety. Okay, which variety you should edit.
So the trait we want to modify.
Yeah. Second, you should take the trait. Okay. First, you choose a variety. Then, by analyzing what is the weakness and what is the opportunity and the threat, you have to decide which trait you want to modify. Okay. Then, for example, there is a one classical example called as Kamala is an edited variety, or in IRA it is one variety called as MTU 1010. MTU 1010, sorry, MTU 1010. Means both actually Kamala actually rise. It's having higher grain number for panic. Higher grain number for panic. Means why they have chosen this trait? There are earlier previous literature so that if you are increasing higher grain number, you are increasing yield without any compromising any of the trait, like without compromising the grain size or any plant nature. Okay, we're improving this without modifying any other trait. So that you have to select the trait. Then okay, you selected the trait. Then after that, what you have to do, what you have to see? First, you choose a variety where you have to do gene editing. Then you have to select the trait, which trait you need to modify. The next thing is
Design the guide RNA.
which gene is to be edited. First variety, then the trait, then which gene is responsible for this trait. There are several genes involved in this trait. You should select a gene in such a way that if you modify this gene, your trait should be changed in a beneficial way. First, after deciding these three things, then you have to proceed for gene editing. Understood? First, you have to select the variety, then you have to select the trait, then you have to select the gene. Then after, when you're having the gene, it will be easy. Just you order the CRISPR, you order directly. You can, where if you give gene sequence, they will give you directly the constructs. Not even there is no need to design your RNA also. If you give gene sequence, they will give you all your construct. Then you have to do your agromaterium transformation. You have to raise the plant. You have to screen the plant. But this is will be easy thing. But the choosing the which variety, which trait, and which gene, that is very very important before genome editing. Understood? This much
Yes,
you
sir.
have to remember. Why you are doing genome editing? What are the different types? What are the different opportunities we have? And what are the limitations? Okay, you are asking something. Hello?
Yesterday, sir, you said about this hemizygous condition, sir.
Yeah, the transgenic. Yeah.
So in mutation, you said they it will introduce some hemizygous conditions, sir. How can it introduce, sir? Because in mutation we are modifying only already present allele. We are modifying it to a new
See, I
allele.
am
That
telling
means
in transgene.
okay, sir.
That I was telling about transgene. You just watched my video. I clearly mentioned when you are doing with the transgene that this condition will arise. Like that's why I was taking example of T1, T0, like T0, T1, T2, T3. Okay. I was discussing about transgene. If you introduce any other transgene into host, then it will create hemizygous in nature. That I was telling. Regarding transgene, I was telling. Okay. It's not like that.
Mutation cannot introduce, no, sir, that condition.
It may introduce sometimes. You can't say. Okay.
Because it only create new alleles for that gene only, sir.
New
It
alleles it will
can't.
create. But what happens sometimes some insertion also happens. We can't say that. But it will introduce new alleles. It's not be hemizygous. Okay? But when you are introducing transgene, definitely you are introducing new loci into the plot. No? But mutation will not create that condition. Like you directly that's why while transgene breeding I have discussed, I was clearly mentioned some transgene breeding.
Okay.
Otherwise, read Primo's book like transgenic development, like how after developing transgene, how to select the transgene. You read Primo's or T.A. Brown book, it will be very clear. Okay?
Sir, T.A. Brown book that you shared, no, sir, that gene editing.
Yeah, that you can use. Another book is there, Genome. I think which book I shared, I don't know. I think Gene Tech. There are
You
two
shared
books of
gene cloning, sir. You shared gene cloning.
Gene cloning. Okay, that book that is not that is for this cloning strategies. That will be very nice. It explained very nicely. There is another book called as there are 3 to 4 series for that. Genome 1, Genome 2, Genome 3 by T.A. Brown or Gene by Levin's Genes. There will be different volumes will come. That is very interesting to read. Gene cloning also very good book, but I think it mentioned there. But genome also it's very good book. Genome by T.A. Brown or Gene Cloning by T.A. Brown, it's very good book. Okay, understood. Like so that I covered all your CRISPR-Cas9 technologies. Tomorrow we will discuss RNAi, what are the different technologies of RNAi, and we'll see a lot of questions asked from RNAi technology. Then we'll discuss epigenetics tomorrow. What is then day after tomorrow we'll jump into forward and reverse genetics. Okay. We'll cover all in one go. I will cover because it's all linked to mutations. Then I will cover tilling. Then RNA-seq also. No, RNA-seq I will cover plus NGS. Okay. Bye bye. Good night.
Sir,
Yeah.
sir, have you prepared PPTs for other crops, sir?
No. I didn't prepare. Okay. I leaving you. Okay. It's like you take as assignment
Okay, sir.
because everything is same. Again, I have to make the PPTs. See, it is
That means
dictating the notes. It's not like a
each
class.
crop, there are 100 pages like that, sir. So,
No, TA. I preferred crop breeding book. I shared na.
yes, sir, in that crop breeding book only, each crop has 100 pages like that, sir.
See, I given a way how to read the crop. Okay, all the things are not important. Like how I covered the rice and maize, in same way how to cover the different crops. Okay,
Okay, sir.
like why I am not
Okay.
giving these notes for why I am not giving all PPT for all crop. See, while I am reading, I am just taking the points which I feel important. I also may miss some points. Okay, that's why when you're reading the entire crop, like entire all the pages and all the information, some additional point you are going to get. For reading my slides, you may guess that you may miss that point. That unfortunately you may get that in the exam. It will be missed for you only. Like it will be lost for you. Okay, that's
Okay,
why
sir.
I'm giving like first I give an idea like how to read the topics. How which area you have to focus. In that you should read all the things. Okay, that's what I am not doing all the crops. All the crops are not necessary also. Rice, wheat, maize, wheat I am going to cover. But pulses you just remember in pigeon pea you remember cytoplasm. And in chickpea you remember some drought release varieties and some of the important trait. Like this for oilseed crop, quality is very important. Like that you should able to judge where in which crop what is important. That is up to
Okay,
you
sir.
actually. You should able
Okay, sir.
to judge which is important for which crop. Okay?
Okay, sir.
Then like that.
Good night, sir.
Good night, sir.
Good night. Good night.